Libert. And in the lab of Claude Libert, we are working on sepsis as a topic. So sepsis is defined as a life-threatening organ dysfunction caused by a dysregulated host response to an infection. According to the latest global estimates of sepsis incidence and mortality, 49 million people are affected yearly, leading to 11 million deaths, which corresponds to 20% of all deaths worldwide. To date, the management of sepsis is supportive rather than curative and essentially relies on antibiotic treatment, hemodynamic stabilization, and support of failing organs. Unfortunately, after decades of research, the pathogenesis of sepsis remained poorly understood, and no successful therapeutic drugs has emerged that has a clear impact on the patient's outcome. So there are many clinical trials which are targeting inflammation or with immune interventions, but they all have failed in sepsis. So we look to sepsis from a different point of view. We see sepsis more as a metabolic disease, because in sepsis, the pathogenesis of sepsis involves tachycardia, fever, tachypnea, immune activation, coagulation. This all requires supraphysiological energy supplies. However, despite their increased energy needs, the septic subjects are usually unwilling or unable to eat, which leads to a negative energy balance. The physiological response to a negative energy balance is well conserved. Some species reduce their metabolic needs by reducing their body temperature and going into a so-called hibernation. And other species, like the mus musculus and humans, activate the starvation response. And the starvation response liberates energy from the body reserves to foresee in its own energy. Two transcription factors play a key role herein, GR and PPAR alpha. So GR, or the glucocorticoid receptor, is a nuclear receptor which regulates glucose metabolism. So via glycogenolysis in the liver, I will look for my pointer. Via glycogenolysis in the liver, glucose can be produced, which then can be used by other organs, for example, the muscle, to foresee in energy via the glycolysis pathway. And glycolysis will lead to lactate. Lactate in turn can be taken up again by the liver and be used again in the gluconeogenesis pathway to produce glucose. It's actually a cycle that we have here, the Cori cycle, with gluconeogenesis and glycolysis. So as said, the glucocorticoid receptor is a nuclear receptor which mediates the action of glucocorticoids in cells. It's not only important for the glucose metabolism, that's also where the name comes from, the gluconeogenesis, the glucose that is produced with the GR. It also mediates vascular function and immune suppression. Glucocorticoids can be produced by your own body, so endogenous glucocorticoid production in day and night rhythms, so via circadian way, but it can also be produced upon stress. There are also many synthetic glucocorticoids, like for example, dexamethasone or inhalators, which are used to suppress inflammation. So glucocorticoids are used widely for many inflammatory diseases. Very simply said, the glucocorticoid receptor is this nuclear receptor. The GR is normally in the cytoplasm. When it gets activated by glucocorticoids, the GR is activated, will translocate to the nucleus, bind onto the DNA, and then lead to transcription of GR responsive genes. Another nuclear factor that is important for the starvation response is PPAR alpha, or peroxisome proliferator activated receptor alpha. And PPAR alpha is bound and activated by fatty acids and has an effect on lipid metabolism. So maybe you can see it better here. So PPAR alpha is important for the beta oxidation of fatty acids. And during normal starvation response, fatty acids are released from the white adipose tissue via a process known as lipolysis. And this is aimed to provide energy by means of beta oxidation. And the fatty acid beta oxidation is a multi-step process that breaks down fatty acids in the mitochondria of the cell to produce acetyl coenzyme A, which then can be used by the TCA cycle to produce ATP, or acetyl coenzyme A can also be used in the ketogenesis pathway to produce ketone bodies, and also those ketone bodies are high energy molecules which can be used by distant organs to foresee in their energy. So we studied the role of GR in sepsis, and those results have recently been published in Cell Metabolism. And we also have a storyline in our research group about PPAR alpha in sepsis, and those results have been published in Embryo Molecular Medicine in 2020. But during this presentation, I will focus on the GR story in sepsis. So to study sepsis, we make use of the cecal ligation and puncture model, or the CLP model. In that model, we anesthetize the mice, then the cecum is exposed. We ligate the cecum so that necrosis can occur. Then with a needle, we make a puncture in the cecum so that cecal content can flux into the peritoneum and cause peritonitis in the mice. Then the cecum is put back again and the wound is closed. After approximately 1 week, we see that the mice succumb due to the infection, so due to the peritonitis that we have introduced via the surgery. As a control, we make use of sham surgery, and with the sham surgery, the mice are anesthetized, but there is no ligation and also no puncturing of the cecum. So those mice are perfectly healthy and viable and do not die upon surgery. So we have shown, but it's also already shown in literature, that the GR is really important to protect against sepsis. When we make use of a GR antagonist, RU486, and when we pre-treat mice with RU486, you can see that in sham mice, this does not lead to any problems. But when RU486 has been injected in mice and followed by CLP surgery, then you can see that there is an increased susceptibility of the mice upon CLP surgery. Similar results are obtained in GR-DIM animals. So GR-DIM animals are animals which have a point mutation in GR signaling pathway and has a less functional GR signaling pathway. And also those mice have an increased susceptibility upon CLP surgery. We also make use of ADX mice. ADX mice are adrenalectomized mice. So those mice have been surgically removed from their adrenals and cannot produce their own glucocorticoids. And also those mice have an increased susceptibility upon CLP surgery. So based on those results, we can see that GR is really important to protect against sepsis. However, when we activate the GR with the GR agonist, dexamethasone, we could not have an increased protection of those mice. We do not see an effect with dexamethasone in our CLP model. And those results resemble very well the results that have been published about glucocorticoids in sepsis. There are many clinical trials evaluating the role of glucocorticoids in sepsis, and in 2018, there were 2 very large clinical trials evaluating the role of glucocorticoids in sepsis. The first trial, the adrenal trial, did not show any effect about glucocorticoids in sepsis, whereas the second trial, the approach trial, could show some effect with glucocorticoids, albeit the biological relevance was also rather small. In general, it's now accepted that we can say that sepsis leads to a glucocorticoid resistance. So our research questions is then to understand the mechanism of the glucocorticoid resistance in sepsis. We want to try to refer to glucocorticoid resistance and also study the implication of this resistance on the sepsis pathology. So we know that glucocorticoids suppress inflammation, and that is why they are so often used for many inflammatory diseases. But they also have a metabolic function. As said, they stimulate the gluconeogenesis. Then to study the glucocorticoid resistance genome-wide in the liver, we make use of following setup. So first we do a sham or a CLP surgery in the mice. 6 hours later, PBS or DEX is injected in the mice. And 2 hours after the injection, we isolate the liver for RNA sequencing and qPCR. And when we plot then all the differentially expressed genes upon DEX injection in sham or CLP surgery, we can see that there is a severely reduced response to DEX and CLP mice when compared to sham mice. So the response to DEX and CLP mice is only 19% of that observed in sham. So maybe it's more easily seen or concepted when we look at one dot here. One dot is one gene. And that gene here, for example, has a log-fold chains of 5, so meaning 2 to the 5, increased expression upon DEX injection and sham condition, where there is almost equal to 0 in CLP mice. So there is a genome-wide glucocorticoid resistance in the liver of septic mice. As a validation, we also did some qPCR on the liver, and here we can see that DEX injection in the sham mice leads to an increased expression of that genes, of the GR responsive genes in sham condition, whereas there is no response upon DEX injection in the CLP mice. Then to study the mechanism of the glucocorticoid resistance, we first looked at the levels of GR in the liver. So with Western blot, we looked at the protein levels of GR in the liver. And then we can see that there is no difference in GR expression between sham and CLP mice. Then we looked at the nuclear translocation capacity of the GR. So the GR, once activated, translocates to the nucleus. And here we can see that upon DEX injection in sham mice, there is indeed an increased accumulation of the GR in the nucleus. And this is also occurring in our CLP mice. So there is nuclear translocation occurring in our CLP mice. And indirectly, this means that the GR is still capable of recognizing dexamethasone. So it recognizes dexamethasone and it can translocate to the nucleus. Once in the nucleus, the GR has to bind onto the DNA to lead to gene transcription. So GR is a nuclear factor. And then we can see something interesting. When DEX is injected in sham mice, we can see that there's an increased accumulation of the GR onto the DNA. However, when DEX is injected in CLP mice, we can see there is no accumulation of GR onto the DNA. So there is a problem with GR binding onto the DNA upon DEX injection. And this could explain the glucocorticoid resistance that we observe in the liver upon CLP surgery. Then for the consequences of that glucocorticoid resistance on the sepsis pathology, as said, we see that sepsis is associated with the starvation response. A normal food intake of mice is about 3 grams per mouse per day. We can see that sham surgery does have some effect. There is some reduced food intake, although there is still a decent amount of food intake in those mice. But the CLP mice are very sick and they are unable or unwilling to eat. So we can see there is no food intake of those mice, even though they have access to the food. Normally, when there is no food intake, this is no problem because you have your starvation response, which foresee in your own energy reserves. And this is shown here with the sham starved group. The starved sham mice are deprived from food, so they do not have access to food. So they have totally no food intake, just as we see with our CLP mice. But normal starved sham mice can keep their glucose levels normal. You can see that glucose levels are normal in the starved sham mice. However, when we look to the glucose levels of our CLP mice, we can see that there is a severe deregulation in the glucose metabolism of the septic mice. Initially, we have a hyperglycemic response, which is followed by a severe hypoglycemia. So this is actually a deadly hypoglycemia that we see here. So the septic mice are unable to keep their glucose levels to a normal level. As the gluconeogenesis pathway is very important to foresee in glucose production, we measured the gluconeogenesis capacity of our septic mice. And therefore, we make use of the pyruvate tolerance test. And with the pyruvate tolerance test, pyruvate is injected IP into the mice. In the normal mice, so with sham mice, we can see that upon pyruvate injection, we can see that there is a glucose increase. So glucose is produced upon the pyruvate injection. When we inject pyruvate in our septic mice, there is almost no glucose produced. As you can see here, it seems that we actually have injected nothing in those mice. There is no glucose response in the septic mice injected with pyruvate. But when we looked into the lactate values of those mice, instead of glucose being produced, we can see that CLP mice injected with pyruvate have increased lactate values. So lactate is accumulating in those mice. So normally in CLP mice, you can see some steady increase in lactate, and this is even more pronounced when pyruvate is injected in the septic mice. Very easy said, we can see that pyruvate can lead to glucose production via the gluconeogenesis pathway, but the gluconeogenesis is failing in our CLP mice, as GR is important for the gluconeogenesis, but GR is failing in septic mice. So instead of glucose being produced in our CLP mice, pyruvate will lead to lactate via the glycolysis. So this leads to an accumulation of lactate in the septic mice. And lactate is actually a very well-known biomarker in sepsis, with higher levels of lactate being predictive for a more severe sepsis lethality. So here you can see the lactate values increasing over time upon CLP surgery. And lactate also correlates negatively with the body temperature of the mice. So the lower the body temperature, the more severe the disease is of the septic animals, the higher their lactate values are. Lactate levels also negatively correlate with the glucose levels. The lower the glucose levels are of the mice, the higher the lactate values. And we see that also in human sepsis patients, that patients with a higher lactate value have a more severe SOFA score. So I have shown you to date up to now that sepsis leads to glucocorticoid resistance. Glucocorticoid resistance as such is not problematic, but in sepsis, this leads to more inflammation, less gluconeogenesis, and less lactate removal. So an increase in lactate. Sepsis also leads to hyperlactatémie, which is a great biomarker in sepsis, although the role of lactate is still controversial. According to some researchers, lactate is something beneficial, as it is important for energy redistribution in the body. However, lactate has also many detrimental effects. And we wondered whether GR is playing a protective role towards the detrimental effects of lactate. And to study that, we introduced glucocorticoid resistance by using different animal models. So by injecting the GR antagonist RU486, or by using ADX mice which do not have their adrenals, or GR animals which have a point mutation in their GR. So all those models have in common that they have a dysfunctional GR signaling pathway. We also introduced hyperlactatémie by injecting lactate into the mice, so an IP injection of lactate. So instead of sepsis, which leads to glucocorticoid resistance and hyperlactatémie, we just look to the, according to us, to the two most important targets in sepsis, to GR resistance and hyperlactatémie, which we introduced via another way than sepsis. And interestingly, when we inject lactate in our GR defective signaling models, when lactate is injected in normal mice, this does not lead to any lethality. However, when lactate is in mice, injected in mice which have been pre-treated with RU486, we can see that there is an increased, well, there's 100% lethality of those mice. So GR is really important to protect against lactate-induced lethality. Similar results are obtained in the GR-DIM animals. Also in wild-type mice, there is no lethality, but when lactate is injected in GR-DIM animals, we can see that there is almost 100% lethality. And also in our ADX mice, we can see that lactate leads to lethality. But interestingly, here, when we revert the ADX phenotype by injecting dexamethasone in the ADX mice, we could prevent the lactate-induced lethality of those ADX mice, which again points towards the really important role of the GR of glucocorticoids to protect against lactate-induced lethality. So when we inject our lactate dose, we have levels up to 20 millimolars, which is seen in the most sick sepsis patients or septic mice. And then in a normal mice, lactate decreases again, it is cleared again in the body. However, when we look to the lactate values upon lactate injection 24 hours after the lactate injection in our GR defective signaling models, we can see that lactate is not cleared in our RU model or GRDIM model and our ADX mice, whereas lactate is back to normal levels in control mice and also in the ADX mice which have been pre-treated with dexamethasone. So remember, those mice also do not succumb upon the infection of an injection of lactate. Then we wondered why those mice die, and as they die very fast, we measure the blood pressure in those mice. So when lactate is injected in normal mice, so mice which have been injected with DMSO, this is no problem. They have a normal blood pressure. However, when lactate is injected in mice which have been pre-treated with RU486 and then with lactate, we can see that there is a deeply deadly hypotension in those mice observed. And the same results can be observed in our GRDIM animals, which have been injected with lactate, also though those have a very low body blood pressure upon lactate injection. One of the main causes of shock is an extended vascular permeability. And we measure the vascular permeability in our mice by injecting feeds dextran IV and then looking to the feeds leakiness in different organs. And then you can see that when lactate is injected in normal mice, this does not lead to any vascular permeability. But when lactate was injected in mice which have been pre-treated with RU486, we can see an increased vascular permeability in all the organs tested. And the same results were obtained in GRDIM animals injected with lactate. We investigated whether the extended vascular permeability in our RU lactate, but also in our GRDIM lactate model, could be attributed to VEGF production. Lactate is known to induce VEGF expression, and VEGF in turn has been described to induce vascular permeability, which is a hallmark in septic subjects. And indeed, when we look to the VEGF levels, we can see that VEGF is increased in our RU lactate model and also in our GRDIM animals. We can see that VEGF is increased. And interestingly, when we inhibit the VEGF signaling pathway with the help of PTK787, which is a VEGF receptor 2 inhibitor, when we administer PTK787 in our RU lactate model, but also in our GRDIM lactate model, we can see that we could reduce the lethality upon lactate injection to a certain degree. The same is observed in our sepsis model when when PTK787 is administered to the CLP mice, we could reduce their sepsis lethality. And this was also associated with reduced vascular permeability in most of the organs tested. So to conclude, I have shown you that when lactate is injected in a normal, healthy mouse, this is no problem because lactate can be cleared, glucose is produced, and the mice are perfectly healthy or viable upon the injection. However, when lactate is injected in mice which have a defect in their GR signaling pathway, this lactate cannot be cleared. There is accumulation of lactate, and high lactate values in combination with the GR signaling defect leads to VEGF production, vascular permeability, hypotension, and eventually death. And this is actually what we also observe in our sepsis model. So when we introduce sepsis, the GR gets dysfunctional. There is glucocorticoid resistance. This, together with the high lactate production, leads to a reduced clearance of lactate, so very high lactate use, which will then lead to VEGF production, vascular permeability, hypotension, and death. So according to us, the real key targets for sepsis are the glucocorticoid resistance or the hyperlactatémia that is observed in the CLP model. We still have some work to do. We want to understand the mechanism of the glucocorticoid resistance in sepsis. We are looking at PTM, so post-translational modifications at the level of GR, which could explain the reduced GR DNA binding upon sepsis, or maybe due to chromatin modification, there is a reduced GR DNA binding. We also still look further into the mechanism of GR to protect against lactate. We know that VEGF is a contributor, but we think that there is more ongoing there. We see that there is an increased production of lactate and sepsis, so GR is important to for the clearance of lactates, but we also have some indications that GR is important to inhibit the increased lactate production in sepsis. We want to validate whether the glucocorticoid resistance in the liver is also occurring in a more biological relevant model, like in the pig sepsis model and also in human sepsis patients. So with the CLP model, we study peritonitis, but we wonder whether this is also occurring in other origins of sepsis, like the pneumonia model. And we have a new research line where we study starvation response in sepsis and how the starvation response gets activated. And with that, I would like to thank you for attention. And also I would like to thank my research team, the group of Professor Claude Libert, and also all the collaborators who helped me with this research. And if you have any questions, I am happy to take them.